Okay, well, it's a couple of minutes past 10, so I guess we'll crack on with this. In the last two of these biophysical techniques or introduction to biophysical techniques seminars, if you have any questions, if you could put them in the Q&A box, and then we'll deal with those at the end. I think that's probably the easiest thing to do. And so, yeah, we've got this talk today and another one next week, and that brings the series to a conclusion. Okay, so what am I going to talk about today? We're going to talk about mass spec, proteomics, and intact mass. So mass spectrometry, what's it good for? Well, it's a very powerful technique. Has become a very important technique in biology over the last few years. It's extremely sensitive. We're working down at the femtomolar level and even lower now, attomolar kind of amounts. The thing to remember, of course, is that we're measuring mass to charge ratio here. We're not measuring mass directly. We're looking at mass to charge. And in all this kind of these experiments and any of the techniques you've heard discussed, good sample preparation is key. Mass spec, you should also remember, is not inherently quantitative. And we'll talk about quantitation later on. And also, of course, different compounds ionized differently. So these last two talks, I'm going to center mainly today, or no, I'm not going to mainly, I'm going to totally center around quantitative and qualitative proteomics on the right-hand side here. And next week, we'll talk about some structural proteomics, HDX, and cross-linking. And that essentially is how the lab at the moment here is divided. Sarah works on the structural stuff, and Sue and Frida work on the quantitative side of things. Okay, so a little bit of history. Mass spec, strong history in Cambridge with J.J. Thomson back at the early part of last century now. Out of his lab, I mean, these are just Nobel Prizes that have been awarded in mass spec over the years. But J.J. Thomson, he recruited, or rather poached, Francis Aston to come to his lab at the Cavendish. And between these guys, out of their lab, there were 7 Nobel Prizes, I think, in the sort of first part of between 1897 and the 1930s. So a hugely productive era in mass spec. And then moving on, Wolfgang Paul, who developed the ion trap in the 60s, he was awarded the prize in 89. And then more recently, John Fenn and Koichi Tanaka for their methods of ionization, electrospray, and molding. But of course, there are many other researchers in the past that we're now running with the techniques they developed. You know, people like Peter Rupstorff in Denmark, Dudley Williams, who's here in the UK, Mickey Barber up in Manchester, over in Klaus Beeman in the States. All these guys working on not only instrumentation, but on methods of looking into the physical processes of how proteins and peptides fragment and how you can measure them. So, you know, there's a huge body of people who've done an incredible amount of work to get us where we are today. So here's some of the first mass spectrometers. I quite like showing this. This is the first commercial mass spectrometer built outside of the United States, and this was built up in Manchester by a company called Metropolitan Vickers, who also built ships and tanks and aeroplanes. And this is in 1946. This is the first one, MS1. I guess as it was International Women's Day last week, sorry. It's good to show this. This is a lady called Anne Metrick, and she's a mathematician at Metropolitan Vickers, and she's operating this instrument. What she's doing is measuring the isotope ratios in uranium hexafluoride. In the side here, there's the samples, and I think she must have been a pretty smart cookie to be allowed loose on this particular instrument. And it should be noticed the MRC also has a strong history with mass spec, as the MRC bought the 4th one of these instruments, MS4. I'm not quite sure where it went actually, but anyway, they were in there. On the right-hand side here, these are the precursors of thermoscientific instruments, the Orbitraps and everything that you know now. These were the Finnegan Matt instruments developed in Germany by Kurt Bruni and Ludendorff back in the 60s. fact, these guys started off in the basement of a hospital and were funded by a shipbuilding company. So you see, mass spec has quite an industrial background to it. Okay, so what we're going to talk about today, this is just a wheel. We'll talk about these sort of sections here and leave this till next week. So the technology, what are we doing? Well, a mass spectrometer has several components. They all have the same kind of components. You've got some kind of inlet, a way of getting your sample into the instrument. You've got an ion source, a mass filter, and a detector. And this mass filter can perform some kind of ion selection, if you like. But it's much analogous to lenses and prisms for moving light around. Well, we're using magnets and electrical fields to move ions around. Okay, but they all have the same kind of basic building blocks. We concentrate here on a chromatographic inlet system, electrospray. But again, they're all basically the same building blocks. So here are a couple of, at the top part of this slide, the two main techniques we have in the lab. I'll focus mainly on what we have at the LMB, okay, rather than what's around everywhere else. There's MALDI, matrix-assisted laser desorption ionization, and here you have your target plate, a metal plate with your sample crystallized on that. In a matrix, so the matrix is in the blue, your sample's in yellow, and you shine a UV laser at that at 337 nanometers, and the energy of that laser is absorbed by the matrix, and that plumes away, and you ionize your analyte by some kind of proton transfer mechanism, okay, and then those ions are accelerated down a flight tube, okay. But essentially here you're ionizing by shining a UV laser, and it vaporizes the sample off the target. Our main approach for peptide and protein analysis is electrospray ionization. And here we have the inlet to the mass spectrometer on the right, and you have a capillary here coming from a liquid chromatograph. Okay, so you have sort of a sheath gap down the outside, and your liquid emerging here. And there is a voltage difference between the end of this needle and the entrance to the mass spectrometer of around about, well, it depends what technique you're using, but say around about 2000 volts, okay? So as the liquid appears here, it's in this electric field, you get this Taylor cone formed, and then the liquid plumes off here. And this is a theory now, this is pretty much what people think happens, but essentially what you have is a droplet with your analyte in there, and that's a charged droplet. If you imagine it as a balloon with lots of charges on the surface, and as it moves towards the entrance, it desolvates, and the charge density builds up on the surface. Let's say as you deflate that balloon, or the bubble gets smaller, the charge density builds up until you get some kind of what they like to term Coulombic explosion, where you end up with your analyte charged entering the mass spectrometer. Okay, so those are the two ways that we have of ionizing the sample and getting it from either a solid phase or a liquid phase into the gas phase inside the instrument. And then the two methods of mass analyzer we have are time of flight instruments or Orbitrap instruments. So time of flight instrument here uses, I mean, if you can imagine it as a fairly simple conceptual technique, in that you simply travel the time it takes an ion to cover a set distance, okay? So this is a reflectron. So here ions emerge from the mass spectrometer here, they're pushed down and reflected back up to the detector, okay? And as the distance is constant and the voltage, so the energy you're giving to it, is constant, this equation collapses such that the mass charge is proportional to the time it takes to cover that distance, okay? So that's a time of flight instrument. And then we have Orbitraps. This is the Orbitrap here. You have a central spindle, which is charged. There's a voltage difference between the outside of the shell here and the central spindle. And as ions are introduced, they're repelled by the shell towards the central spindle and then repelled again. So you get a oscillation built up around that central spindle. So that oscillation frequency is detected in the middle there, and then you get the transient, which is deconvoluted by a Fourier transform into a mass spectrum. Now, beauty of this is a very high resolution technique, which is important to us in mass spec. Okay, so these are the instruments we have in the lab. We have some rather nice instruments. This is the latest one, an Eclipse, and I'll talk about that in a little while. But we have Q-exactive mass specs. These are time of flight instruments where we can we use this predominantly for intact mass. The Synapt is used for hydrogen-deuterium exchange measurements, but we can also use it for native mass spec. And this is the MOLDI, and we're all linked in via a LIMS system, which people who access the facility interact with, and I don't need to talk about the rest of that really. Okay, so why measure protein mass? Well, we'll start by looking at intact mass. Well, it's important really. Any of you guys who are expressing proteins for structural work or to look for interactors or anything like that, it's important to know that you've got the right thing. You know, you want to know that you've cloned the right protein. Is it post-translationally modified? Is there some chemical modification on there? Have you had some processing during expression such that, you know, you've got a ragged termini, be that the N or the C terminus? Okay. And it's an important thing to know. So we have an instrument in the lab, there's the Zevo, but we also have an LCT, a little TOF instrument, which is the only open access instrument in the facility. And it's open to people who like to be trained. So if you have a protein that you'd like to measure, come down and see us, and Frida will show you how to use that instrument. Here's an example of what we're looking at. This is myoglobin, a nice standard. This is the charge state envelope of myoglobin as it's the spectrum as it's measured by the time of flight instrument. And essentially we sort of deconvolute a couple of equations down to here, where we assume that the difference in these peaks is due to one charge. Okay, so if you take these as pairs and feed them into this equation, you can deconvolute that charge state envelope, because we're measuring mass to charge, into a mass. Okay, and so actually you don't need to do this manually. There's a computer program on there that iteratively takes pairs all the way through and gets you an accurate determination of the molecular weight of myoglobin. Okay, here's just an example. This is from a PNAC group where they're looking at a protein they've expressed. Looks a lovely spectrum there, but you can also already see that there's something going on. If you zoom in on one of these charge states, or rather several of these charge states, you can see there's probably at least 4 things going on there. And when we deconvolute that, you can see, you know, there's various masses here caused by either adducts or probably more likely more than one protein there, or a ragged termini. Okay, so it's then important to work out what's going on. It is a sensitive technique. The LCT is not coupled to a chromatograph. It's simply hooked up to an infusion pump. So you've got to be quite careful what buffer you've got it in there. No detergents are the kiss of death to electrospray. So glycerol as well. And you should also not have your buffer in TFA. You get a strong suppression of signal in TFA. But there are these kind of things online, and you can refer back to these slide pack if you want to work out which buffers you should be using. But generally I would stay clear of salt and detergent. If you do have any of those, of course, you can clean them up on zip tips and get your samples into more appropriate buffers: formic acid, methanol, acetonitrile. Again, steer clear of TFA. You get a suppression of signal. You'll only get about 10% of your signal. This is the LCT, which is in the lab. Okay, and the other thing to be, these are a couple of things. I mean, here's PEG. PEG ionizes very nicely and dominates the spectra. Here's your protein actually at this end, but that's becoming difficult to see because it's just PEG is suppressing the signal. And here's the kind of salt clusters you get as well. So, you know, very complicated, impossible to do anything with. You're never going to deconvolute that. Okay, so steer clear of salt, detergent, glycerol. Right, so proteomics. Yeah, in the lab, we focus mainly on bottom-up proteomics. And what is it good for? Well, you know, looking at protein-protein interactions, looking at expression levels in cells, either from a disease or from a drug treatment or from a mutation of some particular pathway. but we generally go for bottom-up proteomics. So you take your sample either from a gel or in solution or on beads. We digest that, reduce everything to peptides, reduce it with trypsin, digest it with trypsin, and analyze it on the mass spec. We introduce all the samples into the instrument via a chromatographic setup. You can't just infuse thousands of peptides. You're not going to see anything, so you need to separate those out. So we use a reverse phase setup to clean up the sample and introduce it slowly into the instrument. We determine the mass and fragmentine spectra. We plug those into a database search engine. Mascot and Andromeda in Max Quant are our preferred search engines, and then we visualize those results in Scaffold and Perseus, and these I'll talk about later on, and then you access them via our LIMS. Okay, so here's the sort of general approach for protein and peptide identification. Here's your protein, triptychically digest it down to peptides, look at it in the instrument. Okay, so this is an MS. Okay, so this is a scan of one scan as the peptides elute from the chromatograph. Okay, so we can take, let's say, this particular ion here at 569. If we were to select that and fragment it, what happens is we get a series of fragments from cleavage of this amide backbone. Okay, and these are determined. The ones that we predominantly get with this technique are B and Y ions, but depending where you cleave around that the peptide bond, you get different C and Z, which you get with electron transfer dissociation. But these kind of nomenclatures were built up by people like Peter Ruppsdorf, and the mechanism that has been worked on by Simon Gaskell and Heidi Wysocki, people like this. So there's a huge body of research around this kind of nomenclature and how things work. But you've got to remember that in the gas phase, things are slightly different. So this is a current theory, that there's a mobile proton theory. So you've got a protonated peptide, and you've got it in the mass spectrometer. You've introduced your ion, you've selected 559, the quadrupole's done that. You've introduced it into a collision cell, you've applied some energy, you've got gas in there. These molecules are flying around, collision-induced dissociation, they bash into the gas and to each other. And this proton, that energy of collision, the proton can move up and down this peptide backbone. And you've got to remember, in the gas phase, these are the nitrogen here is slightly basic, as is this carbonyl group here, so slightly different to what you would normally find. But essentially, if that proton resides on that amide hydrogen and you get a collision, you can get a fragmentation of that bond. Okay, and because you've got a number of these molecules, not just one, you can fragment anywhere along that peptide backbone, so you build up a spectrum. And the kind of thing you get is this. Okay, so here's that particular ion that I showed you in the last slide. We've selected that, and we've fragmented by collisionally induced dissociation. And so if we were to look here at Y11, the difference between Y11 and Y10 is 137, so it's a histidine, and then between Y10 and Y9, 128, so it's a lysine, and then Y9 and Y8, it's 97, so you're looking at a proline. And there are nice characteristic things in this kind of spectra, because you'll notice that the intensity of that Y9 ion is much greater than Y8, and that's because it's a proline. Proline is a secondary amino acid, and things don't fragment very well after that. So you can use that kind of thing as a diagnostic. But essentially, this is a little bit complicated, but what you're normally doing is looking for a ladder of fragmentines here that you can then read off the differences between those fragments and work out a fragmentine spectrum. So what we have, and how we determine this, we have an accurate mass for the precursor ion, and now we have fragmentines, and that's the information that we feed into the database search engine. Okay, so it then carries out a sort of pattern recognition kind of process to marry up the precursor ion mass and the fragmentine data with a peptide sequence. Okay, and so it's a bottom-up approach. So by identifying the peptides, we infer the presence of the protein. Okay, so here's an approach that has been used in the past. It's sort of a brute force purification to look for enzymic activity. This was looking for the Hollow-J junction resolvase. This was done with Steve West's group a few years ago now. And so it's a brute force biochemical approach where they have a reagent which forms this kind of holiday junction, and what they can do is they can purify extracts from cells, and then they can see where the activity to make this cleavage is, and then follow that by a couple of different processes. If you follow this line, then you get MUS81 and EME1, which cleave it in a certain fashion, and then another approach of purification gave you this res-A fragmentation, which is the holiday junction, which cleaves it slightly differently. And by homing in, we used this was essentially a gel CMS approach, where what the guys did was gradually fractionate and purify it, work out the fraction that had their activity, and then we analyzed that fraction to look for potential candidates for the resolvase activity. And this was also done in combination with some yeast experiments. And what was identified was this Gen 1 protein. We actually identified it from a very small number of peptides. It was quite a lucky hit, but it's the Gen 1 that is the holiday resolvase. So that's sort of brute forcing. The other kind of experiment you can do is look for protein-protein interactions, and that's either by having an antibody to your protein, such that you can pull that down and pull down with it any proteins that are interacting, and then we can go through a gel or a solution base. Nowadays we look straight at the beads, or you can do this sort of fishing expedition where you have your protein hooked up to some a GST tag, and then you can go fishing in a lysate for proteins that bind to your bait protein. And this is an approach here which was also done. This is sort of that kind of pull down experiment where we started with hectene-3 cells, purified chromatin, and then an agarose beads with a particular antibody to the complex you're looking at, and pull that down. This was looking for components of polymerase 2 and POL2, and here we get the mediator complex coming down. And actually in this particular experiment, I think it was RECQ5L that was identified as an interactor of this complex. And to visualize that, we can use these to look at these interaction pathways. You can look at Ingenuity, which is a little bit expensive, but also there are other tools out there, David, Cytoscape, this kind of thing. Okay, there are problems with pull down assays like that, of course, because you've got a lot of non-specific binding, a lot of washing required. You can elute perhaps transient interactors that would be interesting to look at, because you have to get rid of lots of detergent and things like this. And you've got to have the correct controls, otherwise you can be led right up the garden path. So yeah, and there's a lot of abundant non-specific binders to these dynabeads and the like. Okay, so one way around that recently has been this BioID approach, where you have a fusion protein with this promiscuous biotin ligase. So if you have a protein of interest, you make a fusion protein with BurA, and then when you incubate the cells with biotin over a period of time, anything that is in proximity to your protein becomes biotinylated. And then you can pull that down with streptavidin and analyze it in the way that I've described before. But here's a you can be quite. This is sort of a bit more targeted approach. You can look for proteins that are transiently interacting, because if they're there for a little bit, hopefully they're going to become biotinylated, and then we'll pull those down and see them. And so there are a number of different approaches to that. There's the classical one that I've just talked about there, the BurA fusion protein, or there are other techniques here. You've got HRP linked to cholera toxin, which then is another way when you introduce peroxide and biotin with RL-azide. Here you get labeling. Or there's the sort of organelle specific one, Apex, where here you can target things specifically to mitochondria and all that kind of stuff. But mainly at the lab at the moment, it's the BurA and the BioID and the Turbo BioID approaches that are being used. And so this is just an example of that BioID application. This was John Shin in Sean's lab. They're looking at vesicle trafficking, and as vesicles bind to to the Golgi via tethers, which then introduce, you get snares on both membranes, which drive fusion. So they're looking at these tethers that bind the vesicles, particularly the Golgins. So what they did was they targeted the Golgins to mitochondria and have different constructs of the Golgins attached to burr A, so they can now look for adapter complexes attached to vesicles. And in this experiment, it was TPC1D23 they identified as I think it's a RAM GTPase, an inactive form here in that family that acts as an effector. Anyway, so the BioID, there's a lot of people doing these kind of experiments now. It gives you a slightly cleaner experiment. Not hugely cleaner, but cleaner, a way of identifying interacting partners. Okay. At some point in all these proteomic experiments, they become quantitative because, you know, once you've done your qualitative ID in the first place, then people want to know, well, how much am I pulling down? You know, what are the levels of this kind of thing? And there are a number. There's a number of different approaches you can take for quantification. Label free, which on the face of it is the easiest. You're not doing anything to your sample. You're looking at the peak area under the peptide, okay, and you're integrating that and getting a value. Or there are other approaches like isotopic labeling, be that SILAC, stable isotope labeling of amino acids in cell culture. There's a metabolic approach, or chemical labeling downstream. They all have their different pros and cons, and I think you need to think about it and work out which is the best approach for your experiment. Okay, they all have intrinsic difficulties or problems, but you need to work out which is the right one for your experiment. So label free, you know, you're looking at two states, as it suggests. You're looking at state A and state B, and you're analyzing them separately. So, okay, here's an extracted ion chromatogram of a particular peptide ion in state A and state B, and then we can work out the area under the curve and work out what the expression level is of those proteins. The disadvantage of this is that you need to run biological replicates and you need to run technical replicates, and so your sample number and time of analysis soon balloons. Okay, so these can become huge experiments very, very quickly. Okay, but the beauty is you're not doing anything with your sample. Now there is an approach called spectral counting, which I would advise you really only to think about if you want a very loose indication of what might be going on. If you take this as being gospel, you will be led straight up the garden path. Okay, so this here is the idea here is that, of course, the more peptides you analyze for a protein, the more abundant it is. Okay, so there's protein abundance index, and then there's this exponentially modified protein abundance index. So essentially it's spectral counting. So if you get more peptides, you've got more of that protein present. Be very careful. Okay, it is available to you, but I would encourage you not to use that. And so the label free here, we've got a nice example here in state A and state B. It's a simple biochemical workflow. Okay, remember, you're not doing anything. You can look at whole proteomes, multiple states, but the sample numbers can become huge. Okay, and you need to have a very good chromatographic system, but we don't really need to worry about that too much because we do have good chromatographic systems. Okay. The other approach is SILAC, which has been popular now over a number of years, and this is introducing stable isotopes of lysine and arginine into your peptide, into your proteins. Okay, and you can use this to do all the classical kind of proteomic investigations, regulation of gene expression, biomarkers, pathways, everything that I spoke about earlier. So it's applicable to all approaches. So what you have here is a HEC-293. You have your light, which would be your normal growth conditions, or you introduce a heavy label, lysine and arginine. You grow the cells up, then you lyse, and you mix 1-to-1. And this is the beauty of the SILAC approach, is that you then downstream, you've done exactly the same thing to your sample. Okay, so you're reducing errors significantly. So you've grown them separately here, but then you mix them, and then everything else is done together. So now we're quantifying at the MS level. It's not the peptide, it's not the fragment ion level, it's at the MS level that you quantify, okay? And we analyze these in max quant. And so here's just an example of that. Here's a total ion chromatogram at the top here. And if we take a slice through that at a particular time, we can see that we have ions here that differ. This is arginine 10, so we're differing by 5 Daltons. And this is lysine 8, so we're differing by 4. So why 10 and 4? Well, these are doubly charged. And this is one of the things that I was mentioning earlier briefly about the beauty of resolution on our Orbitrap instruments, and to be honest, it's very high on the Orbitraps, but even the TOF instruments now are able to resolve the isotopes. So here, this is one peptide. It's a doubly charged peptide. You know that because the distance between the isotopes is half a Dalton. Okay, and why have we got this isotopic pattern here? Well, this isotope here is due to the presence of naturally occurring carbon-13 in the environment, okay? And although, it's only there at 1.1% because we've got a lot of the carbons there, the intensity of that is higher. So we can resolve that. This ion here is known as the monoisotopic ion, and it's made up of the most abundant naturally occurring isotopes, okay? So carbon-12, nitrogen-14, et cetera, okay? Right. And one application, they're particularly keen on doing this over at the mitochondrial biology unit, actually, is using SILAC for everything. They use it for their protein-protein interactions, where if you label with isotopes and then pull down, non-specific interactors are always going to be one-to-one in your background, and so you can look for specific interactors. That's one method of doing it. The disadvantages, the advantages, of course, it's metabolic, it's an accurate relative quantification, and you've done everything the same to your samples. The disadvantage, of course, is, it's limited to cell culture, really, and not all cells, okay? Not all cells work particularly well with this. You can't label humans. Well, you probably could, but I think ethically you'd be on dodgy ground. There are Sarah Zanavan up in Glasgow a few years ago, Kruger sort of developed this SILAC mouse, but these kind of things are very, very expensive experiments. So, it has its advantages and disadvantages. Okay, so another approach of labeling, how are we doing for time? Oh, it's not too bad. Is TMT, tandem mass tagging. And this is, to be honest, I think this is our preferred approach in the lab. Label-free and TMT are our two preferred approaches for labeling in the lab. So what you have is an isobaric tag here. So here's your reactive NH2 group. At the end here, you form your NHS ester with primary amines. This is a balancing region in the middle, and this is your reporter region down here. So what you do is you take your sample, you digest it, and at that point they're all digested separately. You then react with different labeled tags, okay? So these they're all isobarics, they have the same mass, okay? But they have slightly different reporter ion masses, okay? So I'll tell you how that works in a second. So you label your peptides, you mix them. So now, because they're isobaric, when they come off the gradient, the chromatographic gradient, they elute into the mass spectrometer at the same time. So when you fragment these now, you get the reporter ions from all your different samples down in one region, and you can quantify on the intensity of the fragment ions, okay? So here are the reagents. This is a 6-plex. This is showing the 6-plex labels. They now go up to 16. So multiplexing is great, whereas with the SILAC, multiplexing is quite difficult. You've really only got 3 sort of possibilities. Here we can now go up to 16. And you can see, so what happens is here you've reacted at this end. This is your balancer region. You're going to fragment around this bond to give you 126. If you fragment here, 127, okay? And so again, that's just highlighting it here. So you label your samples, mass spec, reporter ions it down this region. So now we look at the different levels and we can quantify on the intensity of those fragment ions, okay? Fantastic. But there is an inherent problem with this technique, and I'll walk you through that now. So here's a HeLa cell, 200 nanograms of a HeLa cell lysate on the Eclipse mass spec. This is a total ion chromatogram. So if we take a slice through that at about 47 minutes, these are the ions that are eluting off. This is a yeast. Actually, why does that cell HeLa cell? That is a HeLa cell. But it's irrelevant. This actually is a standard yeast digest that we're looking at here from a standard that Thermo supply, and it's labeled with an 11-plex TMT reagent. So if we're looking at the ions eluting at 47 minutes, we allow those into the instrument. This is sort of a schematic of the eclipse. If you forget this bit here, essentially the rest of it is a Q-exactive instrument. Okay, all down here is pretty much a Q-exactive. So the samples come in, they're focused through an ion funnel around this flat pole, which gets rid of neutrals, into this segmented quad where you do your ion selection. But initially we let things through to the ion router or the C-trap, where they're injected in and detected here, and that's that initial MS that we get. Okay, so now if we select that 596 ion, and the window that we select around that is about 0.7 Daltons, you'll notice it's not the only thing selected. We use the quadrupole to select that. We move it through into the HCD cell, this ion routing multipole, but it's also the HCD cell, fragment it, and in the Orbitrap, and now here's the fragmentine spectra. So we've got the reporter ions here, and we've got the fragmentines. So what we do with the TMT is that we now use these fragmentines to identify the peptide and the reporter ions to quantify it. Okay? Fantastic. The problem is that if you look before, 596 was not the only ion in that 0.7 Dalton window. So you're going to have reporter ions down here, which are not from this ion. And that causes a compression or suppression of the signal. So while you'll get a readout for quantitative differences, that's not a particularly, or potentially not an accurate value, okay? So there's a method that we have on this new instrument called synchronous precursor ion selection. It's called multi-notch a few years ago, developed by Steve Geige's group, but it's now called SPS. And the way it works is this, okay? So here we have, again, the yeast thing. Now we've got another fragmentine spectra. If we look at this 572 ion, we've detected that in the Orbitrap. We now isolate it. Again, look, there's a number of ions here. We select that 572 with the 0.7 Dalton window, but we now let it through to the ion trap, okay, where we fragment it, okay? This is the beauty of the eclipse. It has not only an Orbitrap detector, it has an ion trap detector, okay? So here's the fragmentine spectra detected or measured in the ion trap, okay? So we can identify the peptide. You'll notice instantly, where have those reporter ions gone? Well, the problem, you can't use an ion trap to look at the reporter ions. The problem is that ion traps suffer inherently for something called the 1/3 rule, or the rule of 3, where you can't trap ions that are 1/3 the master charge of your precursor ion value. Okay, so you'll notice this region down here is blank because we can't trap those ions. But what we can do is select a number of fragmentines, and here we're selecting the top 10 most intense fragmentines. Okay, we move those back, we move those ions from here back into the HCD cell, where they're fragmented again and detected in the Orbitrap. And now when we do that, we see the reporter ions and we get an accurate thing. So what we've done there is select specific fragment ions for that initial precursor ion that we selected, and hopefully we've removed any issues we have with isolating peptides. Okay, so whereas before on some of our Q-exactive instruments you might have seen a change in expression of maybe 2:1, 3:1, here you'll see a more true value. It could be maybe up to, you might have seen 2:1, but actually the true value was more like 8:1. Okay, so we should get a more accurate value. I hope you can see, I'm not sure whether your screens are resolving this, but this is an 11-plex experiment, and you'll say, hang on a minute, there's only 1, 2, 3, 4, 5, 6 there, but actually if you look closely, there's another reporter ion here, 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 and here, and this technique relies heavily on the Orbitrap mass analyzer. You can't do this on time of flight instruments. You can't analyze 16-plex on things like the Bruker TIMS-TOF. I think they claim you can get up to 10, but I would worry. So you need this high resolution to be able to use the 16-plex reagent. Okay. What's mass spec also useful for? Well, it's good for looking at post-translational modifications, be those phosphorylation, methylation, sites of ubiquitination, glycosylation, or GPI anchors. I'm not sure about GPI anchors, to be perfectly honest, but certainly the first 4 here. And the approach is pretty much the same. You digest and you look for a mass difference of 79 in the phospho case. Okay, so here's one of acetylation that we did a few years ago. This is looking at SMC3, and it's acetylated in an echo-on dependent manner, and it forms the cohesion of sister chromatids. Okay, and this was stuff we were doing with Frank Ullman a number of years ago now, and he wanted to look at the acetylation. What we identified was the acetylation of one of these lysines, or at least the one they thought was acetylated. We actually identified that both these lysines were acetylated, and you can work that out from the fragmentine spectra, from the distances here. But also, what's beautiful is that you get this ammonium ion here, okay, at 126. This is an ammonium ion here for acetylated lysine, okay, and that's caused by different internal fragmentation, Y and A ions, okay. The beauty of phosphorylation is we can enrich for it using, because they can be difficult to detect because they're you know, in the much like crosslinks, in the milieu of all the peptides you have there, phosphorylated peptides are lowly represented, and so these can be purified using ion affinity chromatography by gallium ion, titanium dioxide. Titanium dioxide is what we prefer using, but you can enrich for those, and then the signal comes up. And here's an example of looking at phosphorylation. This was looking at how extracellular stimuli affect transcription, particularly LPA. This is using the serum response element or serum response factor as a sort of a model system for transcription, and it's activated by two different routes. This myocardium-related transcription factor, or this ternary complex here, and this is what we were looking at. We were looking at the ternary complex, which is activated by this classical MAP kinase pathway, and the terminal accept here is ERK, which turns out phosphorylates ELK1 at the C terminus, changing its conformation, so releasing its from inhibiting the SRF, and activating it. And so this is the kind of again, fragmentine spectra here, we're looking at phosphorylation of this serine, and it's simply worked out by reading the ladder, okay? So we can work out that the difference here is because it has a phosphate group attached to it, okay? And similarly with ubiquitin, just looking, this is Parkin again, a number of years ago now, looking at ubiquitination of Parkin. You can see the classic sort of ladder species here, and I'll come to show you the scaffold in a minute. But again, we can look for here. We can look at a particular B2 ion which has a mass which is fragmentine here with a glide. So if you digest ubiquitinated proteins with trypsin, you leave a glide on the lysine, okay, and we can take that as a modification, and again simply read it off. So from this we can work out which residues are ubiquitinated, but we can also, by looking at ubiquitin itself, work out what the linkage was. In this case it's K48, okay, so we can work out linkage and ubiquitinated residues. Okay, so we've gone all the way around here. The final thing to look at in the last few minutes is the informatics side of things. Now, we are not bioinformaticians or statisticians, but there are a number of tools that we try and use to help us with that. And I know at the LMB, everybody likes to do their own data analysis. So, you know, we do a certain amount, as much as you want. We prefer not to give raw files out because I think that there lies danger, but you know, that's not unheard of either. So one of the tools we use is Scaffold, and that's primarily looking for interacting proteins. It's a good way of visualizing, QCing your data and seeing what's going on and comparing. So here we have a control and a number of test samples here. You can see different interactions going on. The Scaffold program is really, you only need to download the viewer for this, but you know, you can adjust the number of peptides you want to look at. We set it usually for a minimum of 2 peptides, and of course these threshold values here are affecting your false discovery rate. This is quite low. We usually set the peptides to about 20 and the proteins to about 80, but you can adjust these kind of figures to see, you know, what suits your data set. And all of these fields are sortable. So if you click in the header region here, you can move these around and work out which is let's say specific to the control or your sample. So they're all sortable. And you can dig down. So here, this is the sample level. So here are all the protein IDs that you found. If you click down to the protein level, this next box here, having highlighted a particular protein, it gives you a sort of crude coverage map here. Now, these are all the peptides that have been identified for this E3 ligase. Anything in yellow is a peptide that's identified. In green, it's showing you modified amino acids, okay? The cysteines are modified because we react with iodocetamide, but it gives you a nice coverage, and then you get a sort of a probability score here, and also the Mass Spectrometry confidence scores. So essentially with these, anything above 28 generally is considered a significant hit. And also you can dig down now by clicking on one of the peptides, and you can look at the fragmentation spectra, things that we've been looking at already, and it very nicely labels that for you. But remember always that what we're looking for is the accuracy of this precursor ion mass, and then fragmentation spectra to search the database. And if you come down to the similarity tab, you can look for proteins in different species that are also would match to some of the peptides, not all of them, that you've identified for that protein. So you can identify that protein in different species. Okay, generally we'll send you the link to Proteome Software. It's free to download, the free viewer. You don't need a trial version or anything like that. We have the license for it, and you just use it as a viewer. All the functionality is there. You can use Scaffold to look at some of the quantitative data. We tend not to. Okay. The other tool that we use is Perseus, and we're using Perseus to look at label-free data and TMT data and SILAC as well. And this is from Jürgen Cox's lab. They developed Max Quantum this. So essentially Jürgen his raison d'etre is to initially take away the mass spectrometrist's job by making search engines available to everybody, and now he's killing the bioinformatician by giving everybody this informatics and statistics tool. I can say that because he said it with his tongue in cheek. But Perseus, again, is free to download. You need a PC. It won't run on a Mac. Okay, so you need to have a Windows-based PC to run this. And so, by importing this kind of stuff, you can look at sort of scatter plots. So we can identify, proteins that are standing off, this kind of like multi-scatter plots with all this kind of Pearson correlation factors and all this kind of thing. And you can also look at statistically at volcano plots to look for things that are changing. Okay. So there are plenty of tutorials online for Perseus, and there are also tutorials for Scaffold, but you probably don't need as many for that, as that's really just a visualization tool. But if you're going to use Perseus, I would recommend that you look at some of the online tools for how to do things. It's relatively straightforward to import data into this system, but there are then lots of things that you can do within that to tease out what you want to look at. We can look at. I mean, I've just shown you scatter plots and volcano plots, but it does some principal component things. You can check out various the sort of distribution of your peptides to work out whether if it's a normalized distribution, a Gaussian distribution here, then you can work out whether actually the experiments worked or not, and it gives you heat map kind of things. Also, it's a really nice tool. Some people do this kind of stuff in R, but it's available to you. Okay, so these are the guys down in the lab. This is how it is now. I'm of course leaving at the end of next month, and somebody else will be here. So this may change in terms of what the lab focuses on, because I've had a particular interest in the structural side of things. I would imagine it won't change. I imagine it will carry on. But Sue does all the TMT stuff and the quantitative side of things. Frida is qualitative and the intact mass, and Sarah is heavily focused on structural hydrogen-deuterium exchange, essentially. Okay. So if you have any questions, that would be great. I'm going to stop sharing that. And if you have any questions, type them in the Q&A, and we'll see what we can do. accurate? Okay, this is Wenjun Zhang. How accurate by MS to compare two proteins, PTMs? Do we always need to do big N numbers statistical to draw conclusions if you want? Well, I certainly wouldn't rely on an N equals 1 experiment comparing two proteins and PTMs. But you know, the method, if it depends on which quantitative approach you use. If you're going to use the TMT kind of approach or SILAC, then it is an accurate approach. Okay, so I think if you're going to use label free for this, then yes, I think you would need to do a large number to make sure you got the right kind of data. But the labeling approaches, I think you don't need to use a huge number of samples. Could you comment on copy number counting, such as from purified complex? Oh yeah, okay. Well, so we haven't. This is Wei Jing Hong. So we haven't done too much sort of copy number counting. There are, yes, you can do that within Max Quant. There's this idea of working out copy number from quantitative data. That, to be honest, that isn't something we've done. So I can't really comment on that. Yeah. So Wade asks, is it feasible? Is it a feasible way of working out stoichiometry of unknown components of complexes? Yeah, potentially. Yeah. But I think if we were, you know, there are maybe other approaches you might go down for looking at unknown components of complexes. You know, whether you'd, you know, again, you can even some of the classical stuff would help you with that, and maybe some cross-linking. We're also looking at some in vivo cross-linking methods at the moment as well, which could help to look for unknown components of complexes. Okay, well, thanks for everybody for attending. I hope that was useful. I've probably plowed through a huge amount in a very short amount of time, and you know, we were trying to just show you what you can do with the technique and with a few examples, but it is powerful. And I guess I didn't say at the beginning, but you know, we interact as a lab with about 2/3 of the labs at the LMB. So you know, it's a heavily used facility. Okay, cool.